This is a production of Cornell University.
Hey, yeah, so I work with John-Luc Yannick and Virginia Moore on two different intercropping projects. And intercropping is just when you grow two species together in the same time or space. And there's some cool benefits of intercropping that have been described in literature, including increased biodiversity and environmental service capacity of a system, reduce pest and disease, you spread the risk of like natural disasters and still getting some kind of yield. There's the potential for niche differentiation and the compatibility facilitation effects. But for these to work, there's a need for a lot of agronomic innovations, including planting and harvesting, processing, those types of things. And it also presents a novel environment to breed for, since the species must perform well with another species in the system and not just by itself. So we may need to look at both genotypes when we're breeding for these systems. So intercropping two species together, that can be a cover crop with cash crop, or it can be like what I'm working on with two different cash crops together. And they can be configured in different ways, like in mixtures. Think about like broadcasting just different species of seeds. They can be in alleys with one crop between rows of trees, in rows or in strips. And the way I'm doing it with the intermediate wheatgrass alfalfa is alternating rows. And with the oats and peas, with John-Luc's lab, it's mixed within rows. So the oats and peas are together in the rows, as you can see in the picture. And some of the preliminary results I'm going to show today are for the oat and pea project. But I did want to bring up the intermediate wheatgrass alfalfa intercrop because it's pretty cool as well. And it's also a dual use system. So you can, the farmer has the option, at least a little more option on the markets that they go to, where you can harvest for grain and forage. Yeah, so this oat and pea project included 48 different oat accessions and 12 different pea accessions, and that's over 500 different combinations if we're evaluating them in intercrop. So they're spread across 6 different locations where each possible combination is represented at least in one of these locations. And we had 2 different types of peas, the forage, which is this coming up on the screen? No. Okay, so the forage is on the far, an example of the forage is on the far side, and then a field or grain type pea is the one in the middle. We did this, there's like a difference in morphology, where the field type has a lot more leaf area, it tends to be taller, and the grain type is more compact and has more of its tendrils. So the idea is that these difference in morphology could maybe have an influence in the competitive interactions in an intercrop. And yeah, so this year from New York, here's kind of some box plots of what the biomass looked like when we sampled at 4 different time points throughout the season. And we found that the oat biomass in a plot was a lot higher than what we got for the biomass of the peas, but that there's no difference between the oat biomass on whether it's with a field pea or forage pea, or even in the monoculture plots that we did, where I guess I should explain that a little bit. It's like there's this intercrop with a specific oat accession and a specific pea accession, and then right next to it we had that same oat planted in a monoculture. So what we found is no preliminary, at least, that interaction for biomass was not significant. And then for grain yield, I used mixed linear models and did find that certain genotypes were significant covariants for both the oat, the pea, and the total yield of the plot. I do need to do more analysis on this, but it indicates that there are differences in intercrop compatibility. To evaluate how genotypes perform with any genotype of the other species, we look at the general mixing ability. And for this, I used Bayesian generalized linear regression, which is a multivariate model. So I can look at both the outcome of the oat yield and the pea yield with multiple sources of variation from location, block within, year and location, and the oat accession, the pea accession, and then year by itself. So what we're seeing here is in the top one, it's the oat genotype on the x-axis that genotype effect on that oat's yield, and then the P that it's with, any of the P that it's with, is on the y-axis. So in orange, we see that there are some oat genotypes that have a positive effect on both the oat and the P yield in that plot. And the same, we see that there are some that for P genotypes doing the same thing. I was also curious on the competitive interaction between the specific oat accession and P accession. So here what I did was looked at that oat intercrop and the monoculture right beside it for oat yield, and found that some combinations, the oat yield was higher in intercrop than it was in the monoculture right beside it, and then some combinations that oat had a lower yield than it was in the intercrop. Again, showing that there are differences in these compatibilities. So these plots are the specific combinations from Illinois and New York. So that's why not every combination is shown as a color here. Not every location had these monocultures to compare. But yeah, the reds are like, the more red it is, the lower the oat yield in intercrop was and compared to its monoculture, and then greens are where the intercrop oat yield was higher than its monoculture. Comparing this to like these this general mixing ability idea, I saw that there are P's like maximum, where no matter what it's with, it has a negative effect on that oat yield. And there's some also that are at least more positive. So excited to work more on this. And my acknowledgements, I am very thankful to be in two great labs with John Luke and Jenny. Our field crew for this Oat and Pea project is Peter Hyde. Mirza is doing very similar work with winter oats and peas, and we have a great undergrad, Adrian, who's been with us for a couple summers now. I also got great feedback on this from joint lab meetings with the Soros lab, so thank you for that, and get a lot of field help from their field crew of David and Jason. The Oat and Pea project has a lot of collaborators with Patrick Ewing at the USDA Vermont, Martin and Wolf at University of Alabama, Juan David Alvarez at Illinois, Sia Gutierrez Chacon at University of Wisconsin, Stephen Wilkie in Minnesota, and Melanie Caffey at South Dakota. And then the Moore Lab, our field crew is Julie Hansen, Jamie and Ryan Crawford, and Jesse Chavez. And that Alfalfa Intermediate project is funded by the Alfalfa and Forage Research Program. And part of that project was done previously by someone who graduated with their master's in 2024, Megan Williams, and we have collaborators listed for there as well. So, thanks.
Are there any environmental predictors of these compatibilities?
Are there any environmental factors for the compatibility? I haven't looked into that yet. I do need to calculate genotype A environmental factors, and we can get like climate data, but we haven't yet. So those are future things. Thanks.
I was wondering, for like the one variety where you saw it just kind of decreased the oat yield across the board, are you interested in looking further to try and figure out why like that specific variety is decreasing the yield, or is this kind of outside the scope of your research?
Yeah, so I showed that the 1P, maximum P, had an overall like negative effect on whatever oat it was with. And so am I interested in why that might be? Not right now. Maybe. I'm still figuring out where I'm going with my thesis work. So yeah, more interested in more general like interactions, but yeah, totally. It looks like a forage type, but I didn't see any like difference between like forage and field, so not quite sure what that morphology or what that is. Yeah, like what other phenotypes are we looking at? We took height measurements and emergence counts and maturity notes as well. And then Peter Hyde also flies a drone quite frequently. So We have multispectral data that I'm not working with, but we'll have that to compare as well.
So good afternoon, everyone. So can you hear me? Good afternoon, everyone. My name is Beatrice Konadu, and I'm with the Backla Lab. This afternoon, I'll be talking about exploring trade-offs between cold and drought tolerance in Tripsacum dactyloides. And so we picked these two stresses because drought has been known to be one of the single most limiting factor affecting maize production in the US. And then also by building frost resilient crops, we'll be able to improve yields by 10% and also reduce our reliance on nitrogen by 20%. And then based on research, there are some similar shared pathways between these two stresses. But then there's still a knowledge gap between the trade-offs and then the synergies that exist between these two stresses. And that is where this research comes in. And so it got me thinking about how certain maize relatives are able to adapt to these two stresses, whilst maize doesn't have that flexibility. And so I was thinking, could it be that they possess certain specific genetic and physiological adaptations, or it could be due to modifications that helps them to better express their genes or their metabolic pathways or their genomic structures. And then I settled on Tripsacum because it's been known to be the closest perennial relative of maize with frost tolerance. And then evolutionists believe that about 500,000 years ago, maize and Tripsacum diverged from each other, leading to some loss of functions in maize that are still present in Tripsacum. And so Tripsacum tends to serve as like a reservoir for some good traits that could be used to transform maize. And so the data sets that I intend to use to test my hypothesis include some samples of Tripsacum that were sampled by Denise Kostic and Nick Lepak in our lab. They were sampled across the US and then crossed with each other to generate F1s, and then left to open pollinate to generate F2s. And by observation, we realized that there are some of them that are frost tolerant and others that are drought tolerant. And so we have the right population to do this work. And then also another data set that will be involved in this study is a Tripsacum population panel from a Panan project that was conducted in collaboration with our lab. So to test my first hypothesis, I have 8 hi-fi long read assemblies that I'll be assembling to create a PhG, after which I would use that PhG to impute on hundreds of Tripsacum samples from the Panan project. And by doing that, I intend to perform an environmental GWAS study to identify some drought and cold loci, and then also use a DNA language model to identify some causal variants. So my 8 genotypes that I selected, I tried as much as possible to capture enough diversity within the population the F1 populations that we have. And so I used this MDS plot and then selected my 8 genotypes. And so I have my hi-fi long reads now that I am currently assembling. And for my next hypothesis test on the comparative expression analysis of cold and drought, myself together with Lara Brindisi, who is a postdoc in our lab, have been screening Tripsacum under cold. And so what we've been doing is to have some batches that are cold acclimated first. That is, we move them after planting them, we move them to the cold chamber at 10 degrees Celsius during the day and then 6 degrees Celsius during the night, after which they are moved to the freezer at -6 degrees. And during this time point, we sample tissue for RNA sequencing. And then the non-cold acclimation batch, we just planted them in the greenhouse and then move them straight to the freezer, and also do tissue collection at these time points. Another cool thing that I also did together with my PI this summer was to harvest Tripsacum. And so if you've ever worked with Tripsacum, you would know that it's very tedious to do. They tend to shatter and lose their seeds on the field. So if you want to get enough harvest from your seed, you would have to try certain means that end up being extremely tedious. So we tried out this protocol whereby we cut the head, some Tripsacum seed heads together with the cones, and then replace them in mason jars filled with water. And so initially we were very skeptical about this because we didn't know if it was going to affect the viability of the seeds. And so far, so good. We realized that after performing our germination test, when you dehull the seeds, you have about 54% seeds germinating. However, the hulled seeds tend to have like lower germination rates, which is pretty cool, especially for Tripsacum. And so for my next step, the seeds that were obtained from this summer seed harvest will be used to set up my drought tolerant experiment, which will be conducted in the BTI phenotyping facility. Yep, and so that brings me to the end of my presentation. I would, however, like to thank the Buckler Lab for assisting me during this research. I have bagged a whole lot of people, and I would like to thank them specifically for that because I'm not even done with that. And then also, I would also like to thank my committee members for providing insights into this research, and then also my funding sources, the Schlumberger Foundation for supporting my doctoral journey, and then the USDA ARS for supporting this project. Thank you. Yes.
Thank you, Beatrice. That was a wonderful presentation. I'm curious, I joined after you all set up this population. Could you explain why that specific breeding scheme, the process to generate the F1 and F2s was chosen, and maybe some strengths and weaknesses?
Okay, so with crossing, I said that there were some that were drought tolerant, and then there are some that were freezing tolerant. And so by crossing, you end up crossing some of these traits together. And so moving on to the F2 generation, you would have these traits segregating within the population. And so, yeah, that is why we have the F2 and then the F1 population. You're welcome.
So I saw you collected a lot of your samples from like lower latitudes in the US. Do you find that they grow a bit differently or a bit less when they're put up here in New York State?
I wouldn't say they grow up differently, but then they have, so I know that the lower parts are like much more warmer. And so there are some that collections from that side have proved that they are drought tolerant being here under controlled environment. Yeah. So.
Hi, everyone. Yeah, I'm a third year in Wojtek Plowski's lab, but I did a rotation project with Holly, and this is how it started. So I'm actually going to be speaking about my work with Holly, but at the end, I will just warn you there's going to be an abrupt change. I just have a couple slides about Wojtek's work. So how this study arose, I feel like it really started in my undergrad, like when we would be in the field for long days and we would just talk about anything. And a lot of times we talk about career versus family and the challenges with that. And then also just observing a more diverse graduate student population and faculty population. It really made me wonder like where all of the women go. So then, since I don't have any experience with social science work or just understanding parenthood, I decided to read literature. And so the topics in bold are what really like stood or kept standing out. Parenthood kept standing out, and then also in literature they said that the solution was policy. So what's missing from the literature is that there's nothing on this topic, even in plant science in general, but for sure not plant breeding. And then the studies either have women or men. And then also most of these studies only have like 5 or 8 respondents. And I was able to get 31 respondents, way more than I wanted. And then also, so I originally actually wanted to interview the people that had left the field, but that data is either not there or it's confidential. So it made me realize that I have to collect my own data. So this kind of turned into a qualitative project. And I landed on the question of understanding how care responsibilities and gender have shaped a tenure-track faculty's academic career in plant breeding. So to go over a few definitions, I wanted to broaden only talking about parenthood, and I asked the respondents also about like elder care, caring for another person in their family, and also themselves. Another definition I want to go over is gender norms. So gender norms are the social expectation and rules that dictate how people should behave and express themselves based on their gender. This is usually developed during adolescence and can be influenced over time and also by culture. So to go over the process a little bit, because I'm going to assume that most of you have not done this before. The first step is to develop the project and kind of figure out who I want to interview, what questions I want to ask. So I write up forms, like an interview guide, an email that I would send out to people. And I submit all these forms to the IRB for approval. This is also a tedious process, but once it's approved, then I can actually reach out to the participants. And then I conduct a 1-hour semi-structured interview. So that means that I can start out with one question, and then I can ask follow-up questions that the IRB is not aware of. So the questions or the big topics that I asked about were like, what are your care responsibilities? How do they affect your work environment, and were there any policies involved? And then after I have the interviews, I verify that the recordings match the transcripts. And then after that, I code the transcripts with MaxQDA, which is a software to systematically analyze. I think it's probably the most similar to someone like phenotyping for disease resistance. They come up with a standard first and then kind of go based off of that. And so that's kind of what this is doing here. And eventually, after a few rounds of coding, I develop a code book, and that is used for analysis. And then the criteria to participate in the study was that you had to be a tenure-track faculty. You had to be at an R1 university that I selected, but I'm keeping that confidential. And then you have to be defined as a plant breeder by your institution to reduce my bias. And then just some facts about the participants in the study. There were 31 participants, 11 women, 20 men. And I found that to be pretty representative of the actual plant breeding population out there. And then I interviewed assistant, associate, and full professors. And then I will say that there were 8 participants that did not have any care responsibilities. I had to cut the slide, even though it was very interesting. And then also, this is a huge part of the study, is that people who were like international or foreign born, they said that it was much more difficult. But due to time, I had to cut this slide. And childcare and elder care were the most like common care responsibilities. So to dive into like some of the examples from the interview, my first point is that like upbringing kind of shapes cultural norms. So one example is this professor who is in Asian culture. It's very common to expect that the mother-in-law will come for a few months after birth. So he didn't even think to take any time off or do that. So he expected his mother-in-law and wife And then this other professor, both of his parents worked when he grew up, and so he expected that his wife would never stay home, and him and her would also share the care responsibilities. So I'm just going to talk about a few difficulties, and then I'm going to talk about how they coped with those difficulties. So these were the main difficulties that I saw within the interviews. I will only talk about fieldwork and dual career couples today and give examples. Fieldwork, I think, is very specific to plant breeding because you just are so dependent on so many other factors. There's so many collaborators, and also weather is another big one that makes it kind of difficult. So to give some examples on how plant breeding, fieldwork, and balancing care responsibilities is challenging. When I brought this up in the interviews, I got lots of comments like, this is a nightmare. This is the busiest time of the year, hardest part of my job. But in this example, she said that it was very intense, and her husband were both plant breeders, so they had to be out in the field. And she said that she didn't really start actual field breeding until her kids were like 3 or 6 because it was just so challenging. But as they grew up, she was able to spend more time in the field. And then moving on to discuss a little bit about dual career couples. This one PI says that when he says, oh, I need to take care of the kids, his response is fairly positive, but her response, I mean, yeah, she can get criticized sometimes, even though they both actually tried to share their responsibilities. And then I also just want to note that I had a wide range of participants that had a partner as a stay-at-home mom, all the way to like they were also a plant breeder. And even in the relationships where they really did strive to be equal, I still noticed that the mothers were still handling more of the logistics and management of the children, like making sure that they are going to the dentist every year, or like knowing the names of their doctors or things like that. And then moving into how these PIs coped, the biggest one was mainly family that lived close by. And so, for example, this PI, she really relied heavily on her parents to take care of her child for the first 3 years. And she is an assistant professor, and so she said, I really could not have started without my parents. Like, they helped so much. And then the other thing that was the main form of support was just money, being able to hire a nanny. And so I guess as a grad student, I was surprised at how many people had nannies, especially for like 3 to 6 years. So these nannies really became like a second mom to the children. And then daycare was another huge topic. But in this example, she said that there's zero flexibility with drop-off and pickup times. So she would have to leave, and if she wasn't done with work, she would have to work in the evening, which would be pretty exhausting. And then so to finish up with this, the main points were that care responsibilities are shaped by our upbringing and cultural and gender norms. And then fieldwork and traveling were the biggest challenges. I guess I kind of forgot to mention traveling a bit, but traveling, both men and women, I noticed that they had to reduce their amount of travel. And this was even worse in dual career couples. And then community and financial resources were the main things that helped faculty kind of overcome these difficulties and manage it. And so what? So because of literature, I did ask about policy, and I had that in the interview. And not one faculty had anything good to say about policy. So I really don't think that this is the way to go. I think reasonably it would help it more so if we adjusted our mindset and attitude about care responsibilities in academia. I think another thing that would be big is like changing our attitude, especially towards using the policies. There's a lot of negative attitude towards that. And then one thing that I wish I would have asked more so in the interviews is more so about like your work environment. I think that plays a bigger role than if there's policies or not. And then also just building community and having a support network seemed to be the most helpful for these faculty. And now we're going to make the abrupt change. So, yeah, so I am again in Wojtek Plowski's lab. And so I work on double-stranded breaks in recombination with maize. So recombination is obviously very helpful for plant breeders. It will allow us breeders to stack traits or kind of break traits apart to create new combinations of alleles. And then, so recombination starts with double-stranded breaks and it ends in crossovers. And so as we can see, the double-stranded break distribution is quite uniform, but then crossovers mainly happen at chromosome ends. Oh, I guess you can't see it, but there's roughly 200 to 500 double-stranded breaks. There's so many, but then only 10 to 20 crossovers. Meiosis, and so it made me wonder if we could predict these double-stranded breaks compared to these double-stranded breaks in the middle of the chromosome. And so one of my questions is, can crossover competent DSPs be distinguished from crossover incompetent DSPs? And so I am looking at genomic location, CG and CHG, DNA methylation, and DNA replication origin. My hypothesis is that double-stranded breaks that do result in crossovers have very similar environments. And so I think that DSPs in genic regions and low methylated areas and in early replication regions in S phase are more likely to result in crossovers. So I just wanted to say a little bit about this, but I won't go into any results or anything. So I just want to say thank you to Holly for letting me do this project, and also for the participants for giving me their time to be interviewed, and for them to trust me with their stories and experiences, because I think that is really hard to do. And then for Wojtek, for my double-stranded break project. Thanks. Yep.
I was just wondering, how do you feel about your like two different types of research that you're doing? Like, is there something that you like more or less about the kind of more social research versus what you're doing now, and what motivated that switch?
I guess, so what motivated the switch is I worked in industry for a little bit in between my undergrad and grad school, and I just felt like I had such a horrible experience that it kind of pissed me off, and I said, I'm going to do this project with Holly. And then I guess, I forget what the first part was, but just the differences.
Yeah, but if there's something that you like more about one versus the other?
I will say with the DSB project, it is really hard to analyze ChIP-seq data. Not with like transcription factors, but something that occurs all across the genome that is difficult. And then also something difficult with the qualitative data is that each story is so unique that it's just so hard to kind of make sense of it, which is why I find Max QDA really helpful so that I can kind of mark up the transcripts.
Yeah, I also just want to comment on like kind of how poetic the switch was, talking about like going from like your work to your like life and how that balance sometimes isn't balanced as it kind of was in the presentation. So I really enjoyed the fact that you included them because they're both really important to you. So in the CARE project, is there any sort of continuing thought that you have a conversation that we can start having here at Cornell to be able to, you know, open the dialogue up for like women who want to go into like faculty positions and worry about having children and timing, and like if you have any thoughts.
Yeah, I have thought about potentially having like a discussion here or at a conference, especially with like the generation now and the older generation, to hear some of the challenges that they've gone through. Yeah, because that is actually something that kind of prompted this, like having conversations with older women and just kind of seeing how sad they were. And then also just having conversations with my peers. I feel like a lot of people kind of worry about being able to do both.
Take time off for these kind of responsibilities? Were they similar, or was there a lot of variation depending on what university participants had?
I guess they were similar as in that they had parental leave, but the amount of time was very different. Like some were like 2 weeks, some were 10 weeks. I think at one university it was like 2 months. Oh,
I just want to point out that this afternoon the graduate science are having discussion about these topics with a big red bar from, I think it's from 4:30 to 6. So there are these kind of discussions going on campus. And yeah, that's what we're talking about.
yeah. Thanks.
Thank you, Quinn. As a sociologist in the plant-breeding program, thank you for doing this, considering your partner. You mentioned that when we were explaining the participants, there were a group of people who do not have diversity skills. How do you define that? So that they don't have family, or they have older people to care for, or they don't have children. What do you mean by that? And then my second question is, when you mentioned that for dual couples, there is more toll, it takes more toll on the woman than the man. What exact impact does this have on you?
Yeah, those are good questions. So I guess I'll answer the first question first. So, how did I define someone with no care responsibilities? It was someone that either didn't have any children, didn't have any sort of dependents, and then also people that had not yet had to take care of their parents. I know eventually they might have to, but they didn't really have any sort of care need. They also said that it wasn't really a burden, like they didn't have any health issues, anything self-related. And then, can you remind me what your second question is?
But you were talking about the dual couples.
Yeah.
And I was asking the field work and the traveling to more toll on women than the men. So what exact impact does this have?
Yeah, one example that I had to cut out was that this female faculty, she said that her husband also was a faculty. I mean, he also was a plant breeder, but in industry. And so he had to drive, I think he worked like 20 minutes away. And so she said since she was closer, she still had to, like if there was any sort of emergency, she would have to go pick up her children. Or she would have to take the day off. She was always kind of the one sacrificing if there was any sort of need that the kids had. If that makes sense. Does that make sense? Okay.
This is more a comment versus a question, but one of the side effects of COVID and everything going on in the community is that they're virtual. A lot of these offer a virtual option for people who can't go. So the caregivers who can't leave, you can't work it all. They can actually still tie in for like a small fee if it's supposed to online or so that's something that I don't think organized communities acknowledge that. I don't think they realize that that's actually a lifeline. You can't travel. That's what it's about. So that's one of my comments. I don't know if organized communities are aware of that. They offer it, but
Yeah, that did come up in the interviews where some people were like, wow, I am on Zoom, but I still have to take care of my child. Mhm.
This has been a production of Cornell University, on the web at cornell.edu.